I'm not really going to talk about our own work in the lab. Rather, I'm going to give you a 30,000-foot overview of the interaction between the microbiota and chronic liver disease. I was asked to focus primarily on non-alcoholic fatty liver disease and NASH because I think that this is probably the most relevant to HIV and the drugs that are being used. But I will use as a couple of examples other disease processes. And what I'm going to really try to do over the next 25 minutes or so is try to get this as close to human biology as possible. So I'm going to use some, I think, salient examples of how the microbiome and their metabolites may be influencing chronic liver disease. So this is what the order of my talk, and we've already heard a really nice introduction. I'm going to give you slightly different view of it. So the microbiome, and we'll be hearing about the other types of organisms other than bacteria, but for the purposes of this talk, I'm going to be focusing on the bacteria. We've already heard about the complexity of the gut microbiota in particular. So this is my 30,000-foot view of our interaction with the microbiota. So topologically, obviously, the gut microbiota is in the external world, and in the internal world is the mucosal immune system, obviously oversimplified here. But what separates us from the external environment is remarkably only one cell layer thick, and that's the intestinal epithelium. So we obviously produce substances like mucin and antimicrobial peptides that help to shape the composition of the microbiota and have alter its function. In return, the microbiota interacts with us, producing various substances as well as certain taxa that are more anti-inflammatory, T-regulatory types of responses, other more pro-inflammatory, and a lot of interest now about the ability of the microbiota in general to program plasma cells to secrete specific immunoglobulin A. For the purposes of my talk, it's also very important to recognize that what you eat in terms of diet, antibiotics, and xenobiotics can not only shape the composition of the microbiota, but especially diet can serve as a substrate for the production of various types of small molecules that we end up absorbing. And the notion is that our ability to absorb this myriad of small molecules made by the gut microbiota, which, by the way, I don't think we have a good understanding of how many molecules there are, but a lot of people are studying this, together with the effect on mucosal immunity, we think plays a role in the increasing incidence of diseases that we see associated with industrialization. But I think it's very important to recognize that we have co-evolved with our microbiota largely to live in a mutualistic relationship. This is something that was brought forth by Jeff Gordon probably over a decade ago, in that the host provides a lot of benefits to the bacteria, but then the bacteria provide a lot of benefits to us. And what I've highlighted here are just some interactions that are actually relevant to the liver. So let me now segue to liver disease and focus on NAFLD and NASH, some plausible interactions here. So, you know, what is the relationship between the gut microbiota and the liver, and why is it such an important physiologic target? Well, the reason is obvious. There's a direct portal between the gut and the liver. So the liver receives 75% of its blood supply from the portal vein. And so that means that a lot of things that are produced by the microbiota actually influence the liver. And because of this, there are interactions between the microbiota and many different types of liver diseases. And you could spend an entire session talking about any one of these different types of diseases. But let me just focus on NAFLD and NASH, so non-alcoholic fatty liver disease and non-alcoholic steatohepatitis. So this is sort of a spectrum of disease. It begins as steatosis, just fat in the liver, which is remarkably benign. But for unknown reasons, certain individuals will begin to develop inflammation. And as we begin to see hepatocyte injury associated with inflammation, we develop another entity called non-alcoholic steatohepatitis, or NASH. That then leads to the progression of the disease and fibrosis, ultimately to cirrhosis, and increasing the risk for hepatocellular carcinoma. So NAFLD, just fat in the liver, it's an enormous finding in Western populations. About 25% of individuals in Western populations have fat in the liver, abnormal fat in the liver, or NAFLD. That's approximately 300 million people. And then, as I mentioned, due to some other hit, maybe genetic predisposition, some individuals, a fraction of these individuals, not insignificant group of these individuals, will begin to develop inflammation. Or non-alcoholic steatohepatitis, estimated to 50 to 100 million individuals. And a proportion of those individuals will go on to cirrhosis. And those are the people that we really worry about, is that conversion of inflammation into cirrhosis that increases the risk for hepatocellular carcinoma, ultimately leading to death. And, you know, as hepatitis C virus is ultimately a curable disease now, non-alcoholic steatohepatitis is going to be probably the major indication for liver transplantation. So you know, I'm talking to you about fat in the liver because it's relevant to HIV. Now, we know that there are co-concurrent infections like hepatitis C virus that together with HIV can accelerate liver disease, and that's why we treat both HCV and HIV together. But we do know that HIV is associated with liver disease, and protease inhibitors can lead to insulin resistance and lipodystrophy and fat in the liver. So I show you on the bottom right-hand corner here just a picture of macrovesicular steatosis with minimal inflammation, but the burden of disease, of liver disease leading to death, is actually not insignificant. So we really can't talk about fat in the liver, NAFLD and NASH, without talking about metabolic syndrome, because they're really very closely intertwined. So metabolic syndrome, as you know, is central obesity, hypertension, hypertriglyceridemia, or dyslipidemia, and insulin resistance. So when we think about the interaction with the gut and metabolic syndrome, you can draw out a diagram like this. So things that live in the gut lumen, and I'm going to touch upon each one of these things, flow through intestinal tissue, and it can impact on intestinal tissue and lead to alterations in immunity, as I mentioned, production of gut hormones. There are neuronal inputs that can be affect centrally mediated mechanisms that affect energy balance. But these same types of products can also have an effect on the liver, both inputs leading to alterations in energy balance and then promoting metabolic syndrome. So when we think about the animals, excuse me, the animal models that we have for obesity and metabolic syndrome associated with the gut microbiota, there are a number of different types of animal models here, and they have different types of phenotypes, and you see different types of associations. I'm not really going to get into any one of these things in particular. I just want to give you an overview, and one way of looking at this is that there are some animal models of metabolic syndrome and diet-induced obesity through the microbiota that are diet-dependent. Others deal with small molecules, and short-chain fatty acids are produced from fermentation of undigestible carbohydrates by the microbiota, and it's been a target of a lot of intense interest. And then, as I mentioned, immune function also has an input through the development of metabolic syndrome, and even anatomic. And so there are a number of papers looking at Roux-en-Y gastric bypass, the effect on the microbiota, and functionally what that might mean. Again, mostly in animal models. So I mentioned this on the previous slide. One of the most salient features of the gut microbiota is it's deeply fermentative. So it digests complex carbohydrates into short-chain fatty acids that are used as an energy source and leads to lipogenesis in the liver, and that might be important for NAFLD. But these short-chain fatty acids also have epigenetic effects, and they also activate G protein-coupled receptors. So the whole notion that these molecules that are produced by the microbiota or modified by the microbiota could actually be signaling molecules to receptors is a very important concept that I will be coming back to later on in my talk. Again, with that 30,000-foot view in mind, what are the different types of pathways when we think about the input of the microbiota into metabolic syndrome and obesity? Well, roughly divided, there are microbial metabolites that can either affect epithelial cells themselves and lead to inflammatory processes, but as I mentioned, can induce various types of products like peptide YY, or activate various type of G protein coupled receptors. So there are examples of all of these in the literature, looking at various cell culture model systems, various type of knockout mice, to show that, for example, again, short chain fatty acids will have this effect that ultimately leads to inflammation, metabolic syndrome, and obesity. But the other way of looking at it is really thinking about the direct effect of microbes on immune activation. And again, so there are various models. TLR5, a knockout mouse, is one particular model that leads to dysbiosis and then alters the microbiota that leads to an inflammatory process. So there are many examples of this, but it's not the metabolites, but the microbes themselves that are playing a role in metabolic syndrome. All right, so what about diet? Diet obviously is a major driver of obesity. In fact, I'm a believer that that's the primary reason for the increasing incidence of obesity and metabolic syndrome. So this is the only piece of work that I'm going to show that we published many years ago, and this was an association between all these different types of micronutrients and different bacterial taxa. The positive correlations are in red, the negative correlations are blue, and the intensity of the colors indicates the strength of the correlations. I just want to point out one thing, and that there are two genera here, Bacteroides and Prevotella, that have distinctly opposite associations with micronutrients. And I'll come back to that on my next slide, so just remember Bacteroides and Prevotella. So there are associations between a diet in humans and the microbiome. And in general, we see a decrease in microbiome richness. That's a decrease in the numbers of bacteria in their genes that are associated with both disease states and the consumption of a westernized diet. So what do I mean by that? Individuals with marked obesity, insulin resistance, dyslipidemia, inflammatory phenotype have low bacterial richness. On the other hand, consumption of an agrarian diet is associated with increased bacterial gene richness, and energy-restricted diets increase bacterial gene richness. So this is essentially what we see in individuals living in agrarian cultures. For example, Africa having higher microbiome richness versus individuals living in industrialized nations. And individuals in a more agrarian culture have a decreased incidence of various types of diseases, immunologic diseases, as well as metabolic diseases. That doesn't obviously necessarily mean it's the microbiome that's doing that. Obviously, the microbiome could simply be responding to diet. But let me show you a little bit of controversy, but there is some, I think, an important point when we talk about this. One issue is that how you look at the microbiome sort of is very sensitive to the way you analyze the data. So this is the notion of enterotypes that was put forth by the European MetaHit group. And the idea was that individuals could be classified based on the composition of the microbiota, and they claimed that there were three major drivers of this associations, with Ruminococcus predominant, Bacteroides predominant, and Prevotella predominant. I think there's been a lot of discussion about this, and I think that the best evidence that we have right now is it's more of a gradient, and that at extremes you have Bacteroides predominant and Prevotella predominant individuals. But the interesting thing is that they seem to co-exclude each other in the same environment, and that is individuals that have high Bacteroides have low Prevotella and vice versa. That has been demonstrated time and time again, both computationally as well as in many different data sets. So the interesting thing is that individuals largely living in more agrarian cultures tend to have a higher abundance of Prevotella and lower abundance of Bacteroides. And individuals living more in industrialized nations have just the opposite, higher levels of Bacteroides and lower levels of Prevotella. So why do I tell you about this? Well, there was a paper that was published by Frederick Backhead, which I think was quite interesting. So this is, again, trying to get a little bit closer to humans. So it's actually been known for a very long time that the consumption of fermentable carbohydrates that we can't digest, and in this case, Frederick was looking at barley kernel residues, that seems to improve glucose tolerance through mechanisms that have not been completely well characterized. But in human populations, individuals that have higher levels of Prevotella and lower levels of Bacteroides seem to respond better to this dietary intervention in terms of glucose tolerance. And then he goes on to show that the phenotype is transferable on mice and gets into some mechanisms. The point, I think, is that if this is reproducible, and very robust, it could be a biomarker for individuals that might respond to a certain type of dietary intervention when we think about metabolic disease. The other and more aggressive thought would be, well, maybe you could change somebody's microbiota so they would respond better to dietary interventions. So I think this is where the field is sort of going, and I started with a relatively controversial notion that has evolved in nature in terms of analyses to what we actually observe in human populations, and now maybe to some type of functional relevance to metabolic disease. Again, with the notion that we're trying to get a little bit closer to humans, what do we know about metabolic syndrome and the gut microbiota in terms of transfer? Well, you all know the story about fecal microbiota transplantation, which is exceptionally advantageous in individuals that have recurrent Clostridial difficile infection. A lot of you may also be aware of the work by Max Neudorf, where he transferred the microbiota of lean individuals to individuals that had metabolic syndrome, and could show a statistically significant but modest improvement when he did this. Now he's gone on to expand this cohort and has shown that this continues to be the case. The effects are relatively modest. However, this is very important data because this is human biology. There's a lot of information in animal models, as I've shown you, but this is one of the few examples that in humans that significant alteration of the microbiota using a very complex community approach might have an impact on human biology. So I think we should stay tuned. These are very interesting data sets that certainly bear a lot more of analysis. With that notion in mind, there are a lot of clinical trials ongoing with FMT, looking at many different types of disease states, and I highlighted the ones that are associated with NAFLD, NASH, and obesity and metabolic syndrome. There's a lot of interest in this because it works so well in Clostridial difficile infection. Obviously, that's an infectious process, right? That's not a complex disease entity with a genetic input, and it's not necessarily a chronic disease. So I think the notion that FMT would be advantageous in many types of these diseases, I think waits to be determined, but I would not be terribly optimistic that you would see a very significant input. Nevertheless, I think understanding whether or not you see anything at all is extraordinarily valuable. So the collection of the biospecimens in these data sets, if a subset of individuals respond or if they don't respond, analyzing those data sets tells us something about human biology, because that's really the only approach that we have right now. So now let me talk about the gut microbiome and NAFLD and NASH, again building on some of the concepts that I talked about earlier. So the concept is, again, things that live in the gut can transmit things directly to the liver. But in order to get to the liver, you have to get through the barrier, which I'm sorry, it looks terrible here, but I'll walk you through it. So it turns out that the microbiota obviously makes various types of metabolites, ethanol, trimethylamine, short-chain fatty acids, then flow into the liver and can have an effect on lipogenesis, for example. But then bacterial products, lipopolysaccharide being one example of this, can flow to the liver where it can play a role in inflammation. And there's a notion that LPS itself, through toll-like receptor 4, might be playing a role in progression of cirrhosis or fibrosis in the liver. But then alterations of the host themselves can alter the microbiota, leading to dysbiosis that then accentuates this circular phenomenon. So the issue is that the microbiota can transmit signals to the liver, but the leaky gut is a common theme in a lot of these diseases in which the microbiota is trying to communicate to the host. And that'll be an important concept that I'll come back to towards the end of my talk. All right, so the notion that the microbiota function might be altered with NAFLD and NASH, thereby those taxa themselves or their byproducts could be accentuating disease, is inherent in the notion that the microbiota is different in NAFLD and NASH. And so there are a number of studies that suggest that there is a difference. You can't really read any of this, and that's on purpose because it's not really important that you know that you can't read any of these things, because the effects are relatively modest, at least in my mind, and not completely reproducible between studies. Yet there are some variations. Nevertheless, there are stronger associations with chronic liver disease and a microbiome. And so cirrhosis is one of those areas. Chronic end-stage cirrhosis is associated with a very strong impact on the microbiota as well as the genome of all those organisms, to the degree that discriminatory index can be developed that could be used diagnostically to identify patients with cirrhosis. Now, we obviously have many other ways to identify cirrhosis, but it's proof of concept that the effect is strong enough in certain types of liver disease that you could develop a diagnostic modality. But this will also maybe tell us something about the pathogenesis of disease. All right, and then in my last portion of my talk, let me just talk about bile acids, because I think it's relevant to therapeutic interventions. So we all know that bile acids are produced in the liver from cholesterol into conjugated bile acids, and they're dumped into our intestinal tract, and then they're reabsorbed in the terminal ileum and returned to the liver. So 90% of those bile acids undergo enterohepatic circulation. They're returned to the liver. But that's a highly regulated process, as I will show you. So bile acids are important for dietary fat and vitamin nutrient absorption because it has emulsifying effect. But bile acids also directly affect bacteria. They have profound bacteriostatic effects that are very dependent on a bacteriotaxa. Bile acids are the new hormone. They are signaling molecules. So they are the ligands for nuclear hormone receptors like FXR and G-protein coupled receptors like TGR5. So the notion is that intestinal microbiota could affect the liver by modifying bile acids and altering, for example, FXR signaling. So we know that gut microbiota converts primary to secondary bile acids. I'll show you that in a minute. And then there's evidence that that bile acid production in germ-free mice versus colonized mice is altered. And it's through the activation of FXR. Again, sort of closing the loop on the effect of the microbiota and liver function. And importantly, it's been demonstrated that activation of FXR can improve intestinal barrier function. So again, primary bile acids from the liver are metabolized by the gut microbiota through deconjugation and then dehydroxylation into secondary bile acids that are primarily bile acids that we find in the colon. This is actually quite important in Clostridium difficile infection in ways that I really don't have time to talk about, but these do have very important physiologic impacts on the host. So thinking about the FXR is a nuclear hormone receptor found in many different tissues, but at very high levels in the intestinal epithelium and the terminal ileum, the same cells in which enterohepatic circulation actually occurs. The natural ligand for FXR, the most potent ligand, is chenodeoxycholic acid, the primary bile acid. The activation of FXR in the terminal ileum leads to the production of a soluble mediator called fibroblast growth factor 19. FGF19 flows to the liver, where it then inhibits bile acid production. FXR can also be activated directly in the liver. So the notion is that bile acid activation of FXR shuts down bile acid synthesis. So it's an autoregulatory loop. There is a drug that is currently in development right now that I'll talk about, obeticholic acid, which is a very potent FXR agonist. And the notion is that by shutting down bile acid production in the liver, that might be advantageous in liver disease. And as I mentioned, this whole pathway here could be significantly regulated by the gut microbiota because we know that it is in germ-free versus colonized mice. So what's the notion that activation of FXR is beneficial in the treatment of liver disease, both rodents and humans? Well, there have been a number of studies in chronic liver disease in rodents, and I just show you one of these here, that the use of this obeticholic acid, this derived bile acid, can be beneficial in cirrhosis by decreasing the rate of gut bacterial translocation. It does it through several mechanisms. It decreases bacterial fecal load. There's a partial improvement in dysbiosis. Most importantly, investigators have shown, at least in rodent models, that it improves intestinal barrier function. And all these things together reduce liver fibrogenesis. But we actually see an input in humans, and this is very important. There is no current treatment for non-alcoholic steatohepatitis. So this may be the first treatment for this O-beta-cholic acid. And this was a placebo-controlled trial published in Lancet about 2 years ago, where investigators using this drug showed a significant decrease in inflammation, in fibrosis, and improvement in liver function tests through the use of this. Now, there are some challenges because it somehow alters lipid profiles, but there are very large clinical trials ongoing right now, and there are a number of different types of formulations of these FXR agonists. Again, how the microbiota could interact with this, because the microbiota has an effect on bile acids, it's something that is of significant importance in that field. So again, getting back to my very first slide here, just sort of summing up, what I've shown you is that things that live in the gut lumen, especially the gut microbiota, may be important for NAFLD and NASH. Diet and weight control is important, and I gave you a notion that Prevotella abundance in the microbiota may be an important target. Reversing dysbiosis might be something that we would think about, and I gave you an early notion of how FMT could be a proof of concept that altering the microbiota may be important in metabolic syndrome. Bile acids are important through activation of FXR and other G protein coupled receptors, and then enhancement of barrier function to prevent the transmission of microbes, their products, or their metabolites to the liver might be an important concept, and FXR may be one of those potential targets to enhance barrier function. So that's it, and again, thank you very much for giving me the opportunity to speak to you.